Hi everybody, welcome back to Tutorme Alice. Today we are looking at DNA, the code of life, specifically DNA replication. And this is going to help our grade 12 learners pass that exam. Be sure to stick around. We are going to be getting exam tips to help you guys. We're going to be looking at application questions on how to read, analyze, and answer questions on this section. Remember that our paper or your paper that you're writing is made up of 60% application questions. So these tips and us working through those application questions are going to really help your shoe. So be sure to stick around. But anyway, let's get into it. So what is DNA replication? Now, DNA replication is the process by which the DNA molecule makes an identical copy of itself. So basically, it's as if it's in a photocopying machine and it's making identical copies of itself. Now, when does this occur? This occurs just before cell division. So before meiosis or mitosis, remember these are different types of cell division. This one is creating cells that are genetically identical, and this is creating our gametes. But before either which one, mitosis or meiosis, we are going to have DNA replication take place. And the reason why it does this is because it ensures that the chromosome numbers in the resulting cells that are formed are the appropriate number. So remember, we have multiple phases that are in the cell cycle, and specifically DNA replication happens during the interphase. Remember your mitosis and meiosis, those cell divisions have prophase, metaphase, anaphase, and telophase. I always learned it as IP-MAT. Interphase is first, and then we have cell division, which is prophase, metaphase, anaphase, and telophase. So why does this occur? So what is the importance of us having actual DNA replication take place? It increases the number of chromosomes within the cell to ensure that it can be shared between the cells resulting from cell division. Now, the wording is a little bit misleading in the sense because the chromosome number doesn't actually change, but it's the amount of DNA molecules that change. So instead of it being, for example, 46 chromosomes in a single state, it'll be 46 chromosomes but in a double state. And that's the key thing. It enables the nucleus of the cell to split into two so that each cell has the same number of chromosomes. So whether it's mitosis or meiosis, in mitosis, it ensures that the chromosomes have the same identical genetic makeup, but as long as the cells have the same chromosome number. And the same with meiosis. The genetic makeup will not necessarily be identical. They are all genetically different, but the number of chromosomes in each of the cells will be the same. Again, in mitosis, it allows the cells to be identical to each other as well as the parent cell. And remember, this is because during this type of cell division, it's for repair, it's for growth, it is to replace damaged cells. So we need cells that are identical to the parent. Now, how does this process occur? Now, I have to point out this. The sequence or the format that you're about to see is how we're expecting you to answer it in the paper. There are key phrases, there are key words that have to be included, because if you leave these out, you will lose credits for the section. So the first thing is that the double helix DNA molecule unwinds. Remember, helix, in the last lesson we spoke about this. Helix means that it is a twisted ladder, double helix because there's two strands. The weak hydrogen bonds between the nitrogenous bases, because remember, each strand is held together by the nitrogenous bases or the bonds between the nitrogenous bases. And these weak hydrogen bonds break. I highlighted the word weak because many times we see learners leave out this word weak and it gets marked wrong. Two strands unzip, the two DNA strands unzip or separate. So you can use either which word for in this case, but again, you have to indicate that the two strands are now separating. Each original DNA strand serves as a template. And I think this is the important thing because this is a differing factor or a differing statement when it comes to DNA replication and when it comes to protein synthesis. And when we tackle and we delve into protein synthesis, we will see why. But each original strand serves as a template, and we've got to say that each strand serves as a template to form a new strand by attaching free DNA nucleotides from the nucleoplasm. And I think, again, I highlighted this because often learners just leave out the word free DNA. Remember that there's two types of nucleotides. We have RNA nucleotides and DNA nucleotides. So we've got to be specific as to which nucleotide is coming to attach itself. Each DNA molecule now consists of one original strand, so this one original strand from here, and one complementary strand that was formed from all of these DNA nucleotides coming and attaching to it. And we'll look at a diagram just now. As a result, we now have two genetically identical DNA molecules. So the two strands, they are now, or the two DNA molecules that are formed are now genetically identical. And this entire process is controlled by enzymes, pretty much like every process that is in our bodies. So when we look at this diagram here, so this basically shows you a chromosome unwinding in the sense, like showing us the DNA structure and then the DNA unwinding and all of these things that are happening in between. So let's break it down. So obviously that's our chromosome. And this is the original DNA template. So we need to now create an identical copy of it. So this here is the original, and you can see this original strand comes here, and this original strand goes here. Okay. Here we call it the replication fork because at this point, this is where it is forking. It is now separating into the two. It separates. Here we have one of the enzyme, which is helicase, and this is the enzyme that basically separates the strands. This here is the original DNA strand. A reminder, this is also the original DNA strand. Now, these are free nucleotides. So remember, these free nucleotides, the DNA nucleotides, are going to come and attach to its complementary pair. So if this is adenine, it'll attach to thymine, and so on and so on. Ultimately, what's going to happen is that this molecule that is forming here will have the original strand, and it will have the strand that is complementary. And you'll have the same happening on this side. You can see the original strand, and you can see the complementary strand that is attaching here. And again, these are controlled by enzymes, which is DNA polymerase. This ensures that the nucleotides are attaching to the DNA template or the template strands. So we are going to have a DNA molecule forming here and an identical DNA molecule forming there. So exam tip. Remember, we need to pay attention to the format of how DNA replication occurs, as well as the key words and phrases that must be included. Generally for this question here, how DNA replication occurs, it is your longer question. So that means it is generally 5 to 6 marks for this question because they want you to describe the entire process of DNA replication. So when we look at it, remember, write it out clearly. I always tell my learners to do this. Imagine the person who is marking your paper does not know anything about life sciences. it is going to enable you to explain yourself in a scientific way, making sure that you include all of these scientific terminologies and phrases so that you can get all your credits and all your marks, and to ensure that you're explaining yourself in such a way that the person who's reading it understands what you're actually saying. So make sure you pay attention to the format, this format here, as well as you include the words be, that the two strands unzip or separate, that each strand now serves as the template, that the free DNA nucleotides are attaching. So just pay attention to all of those things. And again, remember your complementary base pairs. Your adenine is always attaching to thymine. Your cytosine is always attaching to guanine. Application question time. So here we got a structure or a diagram showing us the DNA molecule just before cell division. So if it's telling us it's just before cell division, we know that it is during interphase, and this is going to be DNA replication. So first question, name the process. So if it's the instructional word is name, it just basically wants you to give the name, and that's it. Nothing else, just what process is taking place. And again, the clue is in the statement that it is before cell division. And going back a couple of slides, the before cell division is interphase, whereby DNA replication occurs. Now, since we've got that question, remember, you've got to orientate yourself. And luckily, they do have a key, but it's not necessary. If you practice this often enough, we always know A is adenine, G is guanine, T is thymine, and C is cytosine. But always orientate yourself. So remember, the pentagon-looking structures are our sugar molecule. Our dots at the end over there are our phosphate ions, and then all of these on this side would be my nitrogenous bases. This would be the original DNA molecule. This is the original template that's here, and this would be my original template that's here. So this means all of these that are coming in would be my free nucleotides creating my complementary strand. So once we've got that, they say identify parts labeled 3. So if we look at what's labeled 3, we've already identified it, because before we look at the question, orientate yourself, see what you can work out, so that when you get to the questions, it's easy peasy, you know the answers, right? So 3 would be my sugar molecule, 4 would be my phosphate ion. But if you want to be very, very specific, which is generally what they're asking us for when it comes to grade 12 life science application questions, they want you to say for 3, deoxyribose sugar, not just sugar molecule. You got to be specific now, because it's a DNA nucleotide, so it's deoxyribonucleic acid. Then identify the nitrogenous bases labeled 1 and 2. Now, if we see nitrogenous base 1, which is here, right? There is no nucleotide that's coming and attaching to it, and it doesn't have a letter, so we've got to work it out. What did we say? We said this is the original strand, and this is the original strand. So you got to do a little bit of counting here, right? So it's going to be 1, 2, 3, 4, 5. I'm going to go on this side. 1, 2, 3, 4, 5. So if this is cytosine, what does cytosine attach to? It's going to attach to guanine. So on this side, G should be guanine, right? Because remember, this was the original and that was the original, and that two originally were attached to each other. So if this is cytosine, then it will attach to guanine, which should have been on my other side. So 1 is guanine. Now what is 2? So 1, again, you're going to go 1, 2, 3. Then over here, it's going to go 1, 2, 3. Again, original strand, original strand. But what are they asking us for? They're not asking you for the original strand. They're asking you for your complementary base that's coming. So what is on the original strand? It's G. So that means on my original strand on this side would be C, and therefore 2, what's joining it should also be guanine. Let's pop ahead and look at our my memo answers to see if we are right. So we said DNA replication, deoxyribose sugar, phosphate ion, guanine, and guanine. Right, so in the memo, it just says sugar. I'm always on the basis of explain it properly, explain it so that it's always there's not gonna be any room for error or any of that stuff. So my suggestion and what I tell my grade 12 learners, always say the exact structure, exact thing. So we're not gonna have a question whether, oh, can it be accepted or would it be accepted? Because you know for a fact, if you say deoxyribose sugar, it's 100% going to be accepted. Next question, it shows us again DNA replication. Shows us here the original strand, template, temperate. These are our free nucleotides coming attaching, and then it zooms in onto one of the actual nucleotides, right? So it says here, identify molecules W and U. So if it's looking at molecule U, it's showing us the entire thing. So what is this entire structure? This is the DNA molecule. What is molecule W? W is the nucleotide. It's showing us the entire thing. Then they say, identify the parts in W labeled X and Y. So X would be my phosphate ion, and Y would be my sugar portion, which is specifically deoxyribose sugar. Now bond Z. Now if you look at bond Z, again orientate yourself. Bond Z is between the two strands. We are saying this is my nitrogenous base. So obviously that's a nitrogenous base and that's a nitrogenous base. So bond Z is going to be my weak hydrogen bonds. Then nitrogenous base V. If we look at it, what is this nitrogenous base here? Because it's been magnified, it's telling us that this base here is thymine. So therefore, V is going to be adenine, because adenine and thymine are complementary base pairs. Where in the cell does this process take place? So where in the cell does DNA replication take place? Where is our DNA found? Our DNA is found in the nucleus. So it occurs in the nucleus. Name the phase of the cycle where replication takes place. So once again, I always remember IPMAT. PMAT is for cell division. So that's mitosis and meiosis. And I is before it, and before it is when DNA replication takes place. So the phase where this process takes place would be interphase. Popping ahead and looking at our memo answer. Again, you can see over here in this memo, it doesn't accept sugar, but it accepts deoxyribose. And that is why I say always be specific, because it's never going to be a question whether it is going to be accepted or not. Because if you give me the correct biological term or the correct phase, it's not going to be a question. It is going to be accepted. I hope you guys enjoy the lesson, learn something, and if you did, please don't forget to like, subscribe, and hit that bell icon, and also hype this channel up. It really helps with getting exposure out there, and it also allows me to help you understand content better, learn, get the tips, go through the application, and just keep excelling in the subject. I have constantly have learners saying how difficult life sciences is actually, and how they often get demotivated. But when we sit and we actually work through everything and break it down for them, they understand how enjoyable the subject is. And I just want to bring that out to you guys. So I will check you on the flip side. Until next time, stay curious and keep learning.